Important to have this continuous dopaminergic stimulation because L-DOPA, as you will see, it's a very important asset for treating our patients, but furthermore, some problems may occur. But today I'm proposing you to have a short journey into the plasticity, and particularly the bad aspects of plasticity, because plasticity, it's a very important and relevant driving force for brain regeneration, and we'll define further on what plasticity is. But plasticity could be also, let's say, somehow harmful. So I will cover my topic today in 3 steps. First, introducing the idea of plasticity and the concept in a more detailed way, and then to have a look on the basal ganglia and the behavior regulation, and finally to point out what are the pathological changes induced by an aberrant plasticity in Parkinson's disease, as these are the reasons for motor fluctuations and dyskinesias, and not only motor fluctuation and dyskinesia in Parkinson's disease, but also for all types of fluctuations in Parkinson's disease and movement disorders, because we have fluctuations in all respects, as we know. It's not only about motor. We are used to have a dominant look into the motor aspects, but it's the same situation in all domains of Parkinson's disease, and I think you are very aware. And also I should mention that Parkinson's disease is not only a dopaminergic disease, and is not only a motor disease, yeah? It's also non-dopaminergic and a non-motor. It's a very complex disorder.
Thank you.
So coming back to the origin, regarding plasticity, I will mention that, plasticity is an important component of this driving force, which is endogenous defense activity, which basically represents the internal power, let's say, somehow directed and controlled by genetic mechanism to create not only post-lesional regulation, because post-lesional regulation is clear now, somehow, but also to reshape our brain activity. And endogenous activity consists of immediate neuroprotection during post-lesional regulation, and further on, regeneration boosted by plasticity, neurogenesis, and neurotrophicity. So plasticity is an important component, and plasticity is not working alone, it's working always in combination with the other processes in a continuous way. So focusing on plasticity and understanding plasticity in the deepest way would say that this is a concept used to describe the brain's ability to change existing structure, because then this existing structure will be changed due to the pressure, normal pressure of life and environmental stimulus, and also after an injury. An injury will definitely create the effect of post-lesional regulation, as I have mentioned. And then this can be measured and described, this plasticity, in terms of structural changes, from cellular changes, but also up to behavioral changes. So everything what is structural and functional and behavior, it's in the frame of plasticity. So the concept of plasticity is a rather broad one. Also, plasticity means the ability of the brain to restructure after training and practice. This is a more physiological approach. And plasticity mediates the transition from early learning to automatic responding. This is the key of our behavior. That's why we are able to do so many things in an integrated way. To integrate the voluntary activity and behavior with the automatic behavior. The automatic behavior is saving a lot of energy and will give a solution for unpredictable situations. For example, when you are driving nicely the car and the kid will jump in front of the car, then you will put immediately the brake. This is not because it's a voluntary, no, it's an automatic response to an unpredictable situation. Because our behavior is a combination between voluntary and goal-oriented activities and automatic responses. So therefore, plasticity mediates this transition and this very important shift. And plasticity is regulated by genetic programs, and this is a genetical strong regulation, as the histones could be acetylated or methylated. When they are acetylated, there is a very intense plasticity activity. When they are methylated, the plasticity is inhibited. There is a post-lesion, there is a let's say post-genetic, epigenetic mechanism of regulation, very complex. MicroRNA, just to mention them, are very involved in this part, and if you are curious, you will see how many advances have been done in this epigenetic regulation of plasticity by microRNA. There are also important molecules stimulating neurotransmitter like glutamate, BDNF molecules that are stimulating plasticity in this aspect. So this is about plasticity, and I think that this is clear. We fixed the frame, how we understand plasticity, and of course don't forget about the broader concept of neuroregeneration, which is also combined with neurogenesis, which is extremely important, and trophicity. Now, if there is something unclear, please let me know. With me it's very nice and simple, just raise the hand, please tell us again this and that. Is it everything clear for you? Okay? Very well. Then we'll continue. So the basal ganglia and behavior regulation. But it's very interesting is that, you know what is basal ganglia, I will not insist. But basal ganglia have been linked to movement control since very long time. We have the impression that we discovered many things recently. But I can tell you that even in the 19th century, for example, in this very, very nice book, The Function of the Brain, in this book, David Ferrier stated marvelous and described the function of the corpus striatum, saying that the centers of automatic and sub-voluntary integrations are located there. So this is an unbelievable statement in 1876. So it was almost clear what is the function of, corpus striatum, of basal ganglia at that time. And this was a really very interesting aspect. Another important thing is that also plasticity was accepted there, and the concept of plasticity was coined in the 19th century. But apparently a kind of dark age followed for more than 40-50 years, when plasticity was denied, was many aspects were, brain regeneration was completely somehow neglected and even denied, as I have said. But after that, starting with 1949, and particularly with the work of Donald Hebb, plasticity was reconsidered and was back on the stage. So regarding the role of basal ganglia in behaviors, because motor activity is a part of human behavior, by and large. So I think this is important to acknowledge. When I'm talking about behavior, we include here the motor activity and motor behavior. And this motor behavior is very important, and all the behaviors are regulated by basal ganglia. And it's important to understand that particularly striatum, which is a very important part of the whole basal ganglia, striatum receive many inputs, many information coming from all cortical areas. And striatum is working like a filter. It's filtering all this information coming from sensorial area, motor programs, behavioral aspects, and then is amplifying. So it's filtering and amplifying what is actually needed in that moment for the individual to survive. because it's very important when an explosion will occur or when a car is coming to run. Yeah, not to stay and to think what's going on, to dream or to do something else. You will do that when you will be relaxed. So striatum is actually the filter of amplifier of all inputs, selecting what is actually critically necessary to do in that particular moment. So that would be the very important function of striatum. And striatum is capable to select this. And it's also a process of adaptation of striatum along the way. And striatum is using important mechanisms. There are shorter mechanisms like lateral inhibition, membrane properties, which shifts between different states of excitability. And these are very quick mechanisms. You know, also a receptor can be expressed in milliseconds, and the receptor may die in milliseconds. For example, AMPA receptors have a very, very short life sometimes. So things are going quick in the brain sometimes. But we have also a long-term oriented change, which is actually represented by neuroplasticity. And this long-term mechanism, and particularly synaptic plasticity, is involved in this kind of regulation of adaptation of basal ganglia, and particularly striatum, for all these tasks. So this complex regulation by immediate and long-term mechanism can preserve or alter or change, keep or change the processes of affective, cognitive, and motor activities. Altogether, we are describing as behavioral activity, and you know, the task of behavioral selection might be pushed further. That is the philosophy of striatum activity, and I think it's very important to understand it in a very comprehensive and simple way. What is good for? You know, that is very important to understand the function. So all these inputs are coming from large cortical areas, and they are going down to the medium-sized projecting spinal neurons. This is the name and the characteristic of the vast majority of the neurons in striatum. Medium-sized, yes, spiny neurons which are projecting further on. They represent 95%, and they are doing their projections in two ways. And from this point on, I think you are completely with me, because these two ways are the very classical ways described as being the direct way and the indirect way. Now you know. So that I will not insist, because we did a lot of teaching courses, Professor Bajanaro particularly, and I did also a lot of, let's say, of developments in this respect, explaining the structure of these circuitries. But what is important to understand is that the nature of these neurons, it's basically an inhibitory one. They are mediating via GABAergic activity, and basically doesn't matter if they are going directly to globus pallidus part interna, which is the output nuclei of this basal ganglia. The output nuclei is always globus pallidum pars interna there. Doesn't matter if you go directly or indirectly, they will reach, finally, the loop will reach this output nuclei, which by their nature have an inhibitory activity on the thalamocortical neuron and regulating this. You know this very well, I will not insist. The direct pathway, the indirect, direct pathway, indirect pathway with a nice station in the subthalamus nucleus. And we have here, in order to understand easier and more related to the disorder, just to mention that this model, it's already a model very, very well described, not recently, but in the late 80s by Albin. And Albin is the developer of this model, and we did a lot of advances after understanding this particular function of the intrinsic circuitry of striatum, because this is about the intrinsic circuitry, as you will see further on. But to make things easier, I will just tell you that look, in this slide, it's easier to observe, the direct and indirect circuitry. But you should know at the end of the day is that the activation of the direct circuitry, which has a phasic activity, will facilitate the behavior and movement at the end, yes, by releasing the inhibition of the thalamocortical nucleus. I'm putting you just the conclusions because I know that you are knowledgeable. And the other way around, the indirect circuitry activation will decrease the facilitation, will inhibit basically the motor and behavioral activities of this thalamocortical nucleus. So what is now clear is that in Parkinson's disease, when the physiological loop, nigrostriatal circuitry is degenerating and we don't have so many dopaminergic neurons, then what will happen? We'll definitely have less activity in direct circuitry and too much activity in indirect circuitry. Both outcomes are generating a decrease in behavior fluidity, in motor activities. Both of them are doing this. You may see from this nice slide. Is there anyone having unclear ideas about these classic described concepts? Is it clear for you? The activity of direct and indirect circuitry, you should know is definitely very well. Then, in this context, let's say, at the end of 80s, beginning of 90s, we were able to explain motor complications, basically dyskinesia and motor fluctuations, by an imbalance in activity of this direct and indirect circuitry. This imbalance being created basically by the degenerative process and by the therapeutical approach. So this was actually the explanation, and there are many papers. You may have a look and have an historical overview on how was everything perceived then. But although direct and indirect pathways proposed by Albin in the original model, it's valid also now, it's very valid, actually has a limitation because this describes only the intrinsic connection, so the internal connections of basal ganglia. But further on, many layers of complexity have been added in the last 2 decades, in the last 20 years, to this part. And today, it's recognized that basal ganglia are subdivided anatomically into functional domains, having a topographical organized structure in an extrinsic way. So this structure is connected particularly to the cortical area and to many other areas. So therefore we have a structure very well described with a parallel, projecting and partially segregated circuitries for these important functions. Generally for emotional activities, for voluntary activities, so-called goal-oriented activities, and for automatic activities. They are very well described today. So this level of complexity brought us in a very good situation to describe the circuitries. And I will simply mention this, and this is very important to know, these complex layers of new structures. And we know today that, for example, the limbic striatum it's represented by ventromedial structures and is connected with the orbital and ventral prefrontal cortex, and it's basically one of the most important driver of the emotional activities. And of course it's connected with the other circuitries, but as I have said, it's partially segregated. Then dorsomedial striatum, it's in the loop of connection for cognitive processes, so associative network, and then dorsolateral striatum is in the loop of automatic activities or habitual activities, as we'd like to describe, and as they have been described originally, supporting all these type of particular motor, fluid, involuntary aspects. Now, the integrating model of the cortical segregated circuitry brings movement disorder, particularly Parkinson disease, closer to the behavioral disorders. And therefore, we have to understand very well the role of this circuitry. Coming back in a more simplified way, I'm subtracting from this slide the emotional component, and we are going here with the cognitive part, which is actually the action outcome circuitry, which is dedicated for voluntary activity, and we call it, whenever you will hear about action outcome circuitry, I will not describe, I described that last year and I think 2 years ago in our session regarding the behavioral aspects. But whenever you will hear about the action outcome, it's about the voluntary cognitive activity related to dorsomedial striatum. And we have here the stimulus response circuitry, which is actually related to habitual or automatic behavior. So there is a strong link between these two circuitries, and they have a big sense, as I have said. The big sense is that everything what we are learning here by a goal-oriented activity from the moment we are tiny little kids and we are starting to walk, and when we are learning to ride a bicycle or to speak, everything is learned here. And then those programs are shifted by plasticity. So we learn by plasticity. Plasticity will shift this into the automatic circuitry, and we'll use that program whenever it's necessary. Because when you are driving a bicycle or when we are driving a car, you are not thinking all the time to all the components of your motor activity. You are doing in an automatic way. And this makes life very nicely. Otherwise, all our life would be a hell to think every tiny little activity and to perform it on the goal-oriented. In a way, that is the case of a Parkinsonian guy. Yeah, basically, you will see further on. And that this is the machinery of plasticity doing voluntary and automatic activities and having a complementary, continuous, let's say, integration. Basically, many skills can be deconstructed into the sequences as basically every component of our motor activity is, every component of motor activity actually has an ingredient, of a voluntary ingredient and an automatic ingredient. I think we should mention that. Now, as I have said at the beginning, it's very important the capacity of basal ganglia to select between competitive options, because basal ganglia, as I said, is a filter and an amplifier, and they will select any second. Yeah, what component of a certain motor behavior or non-motor behavior would be necessary? A goal-oriented one, a voluntary one, or an automatic one? And therefore, it's a very quick decision performed within the basal ganglia. at the end, you know, all these structures, goal-oriented and habitual-oriented, need to interact somewhere to permit the integration. And this is another important part. I will really pass quick through this part because it's very logical. So we have basically the two systems, and I'll go here to this slide in order to make things more simple. Basically, the goal-oriented loop and the habitual loop should converge somewhere. Usually we have this final common pathway down in the motor neurons generating, and the selection is here. Yeah, we have basically a cognitive goal-oriented loop with the selective activities in the striatum and is in dorsomedial striatum, and here we have the loop for automatic activities in the dorsolateral striatum. Usually, what's the problem in Parkinsonian patients? The original degeneration is located in the dorsolateral striatum, and this is confirmed. It's clearly confirmed by pathological findings and also by, you know, all functional tools that are exploring, for example, by PET. We know that degeneration is there, and what's going on? We are losing the substrate of automatic behavior, and this is ruined. And this is the new, let's say, the new vision regarding Parkinson's disease. And because basal ganglia output, as I have said, the output of basal ganglia from globus pallidus pars interna going to the thalamic neuron, it's always an inhibitory one. Yeah? The consequence of this imbalance in the two pathways is usually the increased inhibition and abnormal oscillation, which is generating the motor complications. You know, I try to make it very simple for you. I hope you got the idea. Yeah? So the imbalance in the voluntary and automatic circuitry will generate, by the nature, due to the nature of the inhibitory outputs of the nucleus of globus pallidus pars interna to thalamus, will generate this increased inhibition and abnormal oscillation. I have shown you at the beginning the slides. The idea would be that today there is a tendency to explain many features of bradykinesia, you know, like reduced amplitude of agonist muscles recruitment or ballistic movements, and the rapid decline of repetitive movement by this mechanism. And I think it's a good trend. It's a very new and actual trend. And now, the last part of my presentation will point towards the molecular substrate of the mechanism that are creating this imbalance. Basically, why we have this motor fluctuation and how plasticity, pathological plasticity, is contributing to the development of these complex disturbances. We know very well that L-DOPA is one of the most efficacious drugs to treat Parkinson's disease, but according to the general accepted view, the gradual development and persistence of motor complications are determined by the aberrant form of plasticity, which is triggered on one hand by the degenerative process itself, by the striatal denervation. We are losing more and more dopaminergic neurons, and on the other hand by pharmacological replacement, which is usually discontinuous. And this discontinuity, this unnatural way to stimulate the circuitries, which in natural way it's continuously, and that's why you had the presentation before, with the effort to create a continuous dopaminergic stimulation. That is the sense at the end of the day. Because exactly the discontinuity in dopaminergic stimulation is creating the aberrant plasticity and generating the motor complication and imbalance in those two circuitries. So the brain plasticity potential is very different among individuals depending on the age and genetics, and this is somehow the explanation why we have such an individual profile, such a big variability in different